All right, good morning. So happy Wednesday. What you guys are going to do is finish up the GMO observation questions if you didn't get those done in class. Those are things to think about, like for while the PCR was running, some of the things that are happening and what some of the molecules are that are being used and how they're being used in that process. Okay, remember that GMOs are the genetically modified organisms that you're testing, the foods, and that you're doing this process of PCR, where PCR is the copying of the DNA. So you need dNTPs, which are the loose nucleotides. You need the polymerase, which is copying the DNA, and you need the primers so that the DNA polymerase has something to stick to before it does the copying, okay? And then you have your initial DNA. And every time it copies, it doubles. So, like when we did our human cells, we got that 8 number because we did 2 to the 23rd was our 8 million. You're now going, this one does 35 cycles. So you can figure out how much copies you get by going 2 to the 35, okay? The other thing that you're going to do is to take a look at bacteria and viruses. With that, it's more about understanding what the two organisms are, the differences in them a little bit structurally. Bacteria do DNA replication. They do it a little differently because they have a circular piece of DNA, so they just do one copy instead of having multiple forks at the same time. But then they have ribosomes, so they still are then going to do transcription and translation. What's interesting is that they can actually do their transcription right at the same time as they're doing, then it could be start getting translated right away because it doesn't have to leave the nucleus there. So essentially what you guys are going to do is watch a couple of videos. One is to compare bacteria to viruses. What you'll see here is that viruses really aren't considered living things. They'll have some characteristics, but their reproduction is quite different. They have to have a host cell to reproduce. They can't live on their own. And then there are some things that we're finding as researchers that they can use viruses for. And it's actually interesting that you do still have viral DNA in you. It's played a role in evolution. So that'll be kind of interesting for you to see. It then looks at some of the ways that these organisms get genetic variation because bacteria are going to reproduce asexually. They just do a process of division called binary fission. One divides into two. And then some other information about viruses and the way that they infect cells. And then these retroviruses, and retro is like to go backwards. So retroviruses actually start with RNA as their genetic material. And then they have a protein in them called reverse transcriptase. So they take the RNA and reverse transcriptase, then makes it into DNA using the host cell's information, nucleotides and stuff, and then it then has the DNA that it can embed into the host cell. So those are kind of cool viruses biologically, but they're ones that are around long term. HIV is a retrovirus, and that's why once people are infected with it, they have it. It gets incorporated into the host cells. So hopefully those you'll find the videos interesting. And then there is a knowledge probe just to kind of do a quick look at some of the basics about bacteria and a little bit of basics about viruses. So have a great day, and I'll see you in class the next time.